Hello and welcome to our webinar on Teaching Cambridge IGCSE Biology with David Martindill. I'm Tamsin Hart from Cambridge University Press, and I'll be hosting this webinar for our speaker, David. While we wait for more attendees to join us, let me share some details about the webinar. We're recording the webinar to share with you afterwards, so don't worry if your internet drops out during the webinar itself. If you're having trouble with sound, we suggest using headphones or checking that your speaker isn't muted in your system properties. Your microphones will be muted throughout the webinar. If you have any technical queries or you'd like to say hello and let us know where you're from, please use the chat box. We'd love to hear from you. To find the chat box and Q&A box, please hover your mouse at the bottom or top of the screen and they'll appear. If you lose connection or the presentation at any point, try leaving the webinar and rejoining. Don't worry if you miss anything. Remember, we'll be recording this session today. A week after the webinar, we'll send you an email thanking you for your attendance today with a link to the recording. I'm afraid we're unable to provide certificates, but you can use this email as evidence of your attendance at the webinar, so I would encourage you to do that. Now on to our webinar and speaker, David. Today, you're attending a webinar on Teaching Cambridge IGCSE Biology with our speaker, David Martindill, part of the author team for Cambridge IGCSE Biology Digital Teachers Resource. Our webinar will cover the following topics: how to teach to the syllabus, how our science resources can support your teaching, how to develop your students' essential science skills, how to adapt when you have to teach remotely. David is a practicing teacher who hosts workshops for teachers and school leaders around the world. He has written teaching and learning materials for use in international settings and offers regular online courses for international teachers. David has also contributed to the design and delivery of national education reform programs. He holds a PhD in molecular biology and a master's in education from the University of Cambridge, specialising in practical science. That's all from me. Thank you very much for joining the webinar. I'll join you again for the Q&A at the end. I'm just going to hand over to David now, whose webcam is going to remain off for this session. We're having some issues with that today, but you'll hear him loud and clear. Thank you.
Thank you very much, Tamsin, and welcome everybody, wherever you are in the world. Good morning, good afternoon, good evening, or good night. My name, as Tamsin said, is David Martindill. My background is in molecular biology, but I'm sure, like many of you, upon entering the world of teaching, we've had to broaden our interests. I'm the head of science at a school here in Singapore, so I speak to you at around about 5 o'clock this evening. I'm very fortunate in that I was approached several years ago to get involved with the update for the teaching and learning materials for Cambridge University Press. This was supplementary to some other work I have undertaken for Cambridge, and I very much enjoyed the process, and I'm delighted to be able to share some of the resources with you on screen today. So we're going to try something a little bit interactive to begin with, because I don't want this to simply be a monologue. I'm going to show you the 4 objectives for today's short screencast. I'd like you please to attempt in the chat box to write down what you feel the missing words might be. You don't have to write down all 4, but if you could write down please 1 or 2, starting your suggestion with the letter that shows the missing word. It will be wonderful to see if your predictions for today's webinar are accurate, and it may even encourage me to edit and change 1 or 2 of the slides in keeping with your expectations. So I'll wait for a couple of seconds, and I wonder whether Tamsin, you would be happy in a moment, please, to let me know if there are any common responses that are appearing in the chat box.
We've got a lot coming through already. We've got for A, interesting, interactive, efficient, effective, engaging, active learning, more interactive. And for C, we've got design, and C, to provide. B, we've got formative. C, we've also got teach. Lots of interesting and interactive and engaging for A, and effective We've got engaging for A, online for B, C, prepare, or formative for B. We've got plan for C, but lots coming through there.
You haven't mentioned any D's yet. Has anyone suggested a D as we go?
Let's see. Oh, yes, we have a D, practical, D, learning, D, project, and more learning. That's a theme coming through, practical, interactive.
Superb. Okay, well, I think I've got a very good flavour. Thank you for that, Tamsin, and thank you everybody for your submissions. It sounds to me like you have accurately predicted what we'll be doing today, which is wonderful. That is the objectives list there. However, I'm pleased to suggest we will be able to take at least number 4 in a different direction based upon some of the things you've suggested as well. So we are going to be looking at active learning, formative assessment, planning lessons, particularly using the new Cambridge University Press publications. And we are going to explore briefly how we can engage students with practical led work at home, because I am aware that many countries around the world are still hosting schools that are unable to attend normally at present. So let's get started, everybody. Thank you for that. I enjoyed that moment of interaction. The syllabus, as you know, for Cambridge IGCSE is about to change. That is the reason that the Cambridge University Press resources have been rewritten, revised. So the syllabus, I'm sure you all understand, contains really everything you need to know about the IGCSE course, including the content that students would be expected to master, how they will be assessed at the end of the 2 years, usually 2 years. There's plenty of extra information in the appendix, which teachers and learners often overlook. We are going to be considering the syllabus as we go, because for me, and I'm sure you, it really is the backbone to the entire experience. It's something which is often referred to by both teachers and students as the course goes through. Make sure, however, you check it for the correct year. So if you've got good eyesight, you will see that this one says exams in 2023 to 2025. So students who are about to begin this qualification, ordinarily, if they're on a 2-year programme, would be following this syllabus from now on. Before we get started with some ideas for teaching and learning, I just want to mention one thing, and that is this. You may only pick up 1 or 2 little things from this screencast today, but 1 or 2 little things over time can make a big difference to your practice. The analogy here is in relation to the Tour de France, which is a bicycle race, as we know. Little changes, perhaps to the mass of the bike or the type of rubber on the tyres, they might not add up to much by themselves, but over time, as teachers, if we are willing to make little adjustments to our practice, it does have long-term effects, hopefully positive rather than negative. And of course, I'll only be sharing with you today some of the techniques that I know work. So let's have a moment talking about active learning. I want you to have a quick look at these 2 pictures and just for a moment think to yourself, which classroom is better? Well, that's in many ways an unanswerable question, because on the right-hand side, this would be a better context, of course, for sitting an examination, which is perhaps a summative assessment. So the question really is, which classroom is better for learning? And this links with an age-old dichotomy that people often refer to in relation to what teachers should be doing in the classroom. Should teachers approach students as empty buckets into which we pile and pour knowledge, or should we as teachers be watering cans that help the plants, that is our charges, our students, to grow by themselves? A traditional model is as follows, and I'm sure that lecture-based format is familiar to you. That's how universities quite often still deliver knowledge to students. But active learning in the recent decades has been shown to have enormously significant and positive results on students' progress. I'm not going to spend too long talking about the theory today. We're going to look at practical scenarios for this. But active learning essentially means that learners should do something in lessons. And the evidence suggests that in the long term, students who have engaged in active techniques make the biggest learning gains. So when it comes to simple retention of knowledge, whether they can recall key terms, and there are many of those in biology, as we know, when it comes to things like that, those activities which are more active, which require students to engage with each other, which require students to do something rather than just listen, have been proven with many different types of student to make the biggest gains to their learning. So really, I think our job should be both. We have to spend some time giving students knowledge, but we have to provide some time to let them learn using the tools you provide them. So I just want to have a very quick bit of interaction with you again. Very, very quickly, this one. I'd like you to type in simply one number. Which activity of the following four do you think would be most active in line with the definition I just gave you? We'll just give it a moment, but perhaps again, Tamsin, would you let me know which number seems to be the most common submission?
Yes, I will. It seems to be resoundingly 3, although we have a couple of 4s coming through as well, but predominantly 3.
Thanks, Tamsin. Yeah, but by all means, I think 3 and 4 sound very active. There's a little bit of a red herring, or a little bit of a trick one here. I mean, number 2, by the definition I mentioned a minute ago, is active. Students are doing something. You are not involved. You are not telling them to listen to you if they're sitting doing an exam. So we have to perhaps be very clear about the type of activities we are talking about. It's activities where students are engaging with each other. And I'm pleased to hear that most of you recognize that 3 and 4 are such activities. And we'll be speaking about those now. If active learning is new to you, do start small. Try little activities that last just 5 minutes to get going. My favorites are listed here. So think, pair, share, of which there are many examples in the Cambridge University Press materials I'll show you shortly. Simply ask a question sometimes of your class, give them approximately 10 to 20 seconds to think by themselves, then discuss their thoughts with their neighbour, socially distanced in mind, and then share their ideas as a pair back with you. What's the question is a very nice little activity. Instead of asking a question and requiring your students to give you the answer, why not give them the answer and ask them to design the question? That is a far more active, minds-on means of encouraging students to progress. So in biology for the IGCSE, I could, for example, say to you, what would be the question if CO2 is the answer? And I'm sure sitting there you can think of at least 2 or 3 questions for which CO2 is the answer. I'll give you one of mine. It would be something like, what is the gaseous waste product of aerobic respiration? Yeah. And you could extend this, of course, by insisting on students giving you the command term as well. And that would probably be state. So state, the gaseous waste product of respiration. Converting information from text into a picture is nice, particularly given that biology is an incredibly visual subject, and we are encouraged as biologists to make observations all the time. So instead of perhaps asking students to write notes, get them to summarize a lesson as a diagram, and they can liaise, they can discuss with each other about how they make the decisions as to how to make that into a diagram. And one of my most recent favorites is summarizing the content of a lesson in a tweet. encouraging students to be actively focused on reducing waffle and wordiness and making their thinking concise. It's a real skill in science that we often see students struggle through the IGCSE. Okay, so on to here the Cambridge University Press materials, which have been recently updated, as I've said. I'd like to just take you through a few sample pages to show you how active learning is supported by them. We've got here the course book, the workbook, both of which are written by Mary and Jeff Jones. I'm sure many of you have heard of them. They are the superstars of biology. Matthew Broderick has been involved in both the practical workbook and English language skills workbook, Tim Chadwick and Gemma Young with the maths. My contribution was this one, which I'm showing you nice and big because I'm so proud of it. This is my digital teacher's resource, and I'm very proud of this, actually. I took great pride in writing this, and I'm sure that when you see some samples of the work, you'll see some of the ideas I've already mentioned so far today will feature. So here is a sample spread from the course book. How could we use this sample spread to help active learning in our classes? Well, we have to be a little bit imaginative. There are some very explicit means by which we can engage students actively. So here it says literally, with a partner, think about the answers to these questions. So students are encouraged immediately to engage with somebody else, discuss, vocalize, verbalize their ideas. But then we've got a nice interesting story here, putting the science of cells into context. And I think these contextual starters, which you'll see at the beginning of each chapter, are a fantastic source of material which can be used to help engage students actively. So there are some discussion questions as usual, but you could get them to go away and spend 5 minutes researching some of the key terms that are on the spread here, maybe online, maybe using other textbooks. They aren't just listening to you telling them what they need to know. You'll see on the right hand side here, we've got lots of diagrams. You could, of course, ask students, as I suggested a minute ago, to convert these diagrams into text. You could select a few keywords and ask students to write questions for those. All of these things I have selected today because they can be delivered remotely. Yes, when we all get back to school normally and we can engage in touch and exchanging items on desks, we can start building posters, we can start engaging with extra practical techniques. But even in my school, which remains thankfully open, we are restricted in what we can allow students to do and how they can interact. The workbook here contains lots of opportunities to engage actively with skills such as measurement, such as drawing, calculating magnifications. As you can see, it's laid out in a very attractive way. And I think for students, being able to have a resource like this immediately engages them in active learning through the instructions it has in front of them. So the teacher's resource, which I mentioned earlier, it's a very similar format for every one of the three sciences. You'll see that there are misconceptions listed. And for some of these misconceptions, for example, here, cell structure, there are useful means by which you can, as a teacher, facilitate students overcoming them. So here we've got a misconception, which is very common, that learners confuse the location and the function of the cell membrane and the cell wall. And there is a nice little activity here which requires students to blow up a balloon inside a plastic box and then draw an analogy and comparison between what the balloon is and what the plastic box is in terms of its physical features. Activities are listed which include making cells using just balls of scrunched up paper, drawing the organelles on the inside, and then exchanging them with each other to attempt to identify what type of cell they are. All of these I would assume that you can appreciate are very active, very, very active techniques. students are learning not because they are simply being told facts, but because you have provided them with the raw materials and the tools to make their own meaning of the science. Okay, so we're now going to move on to the second part of this short screencast, which is formative assessment. And again, we're going to have a similar type of format to this part of the screencast. I'm going to talk to you a little bit about what formative assessment is, and then we'll have a little bit of a deep dive into the resources to find out what they can offer. So many of you did write down formative in the missing word exercise earlier. So I don't think this is going to be new to many of you, but just a very quick reminder. Summative assessment is what we do at the end of a topic, at the end of the course. Cambridge International, the exam board, will undertake summative assessment. No comments are ever written on the exams sat by students, of course, because most students will never see them. Formative assessment is what we do on a day-to-day basis. Often this is never recorded on paper. So it's fleeting, it's brief, it's temporary, it's meant to help students reach the next level of their learning. The idea here is that it's ongoing. There's lots of evidence to suggest that if we as teachers provide students simply with summative information about their work, it can have little impact on their future progress, or sometimes even no or negative impact on their future progress. It can be quite disheartening if a student is provided a poor grade, for example. Now, I want to draw your attention to the very last row here. If you give a student of yours a comment, a written comment, it could be an abbreviation, we'll talk about those in a moment, but more than a comment, a suggestion as to how they could improve, or maybe another question, then that can have a very strong positive impact. It can foster an even closer teacher-learner relationship as well. Feedback really should be positive. Look for the good in every piece of work that students provide. It should tell learners where they are in their learning. And what that means is not necessarily a grade, but it should reveal to them whether you feel they are on the right path. And more than anything else, it should give them a hint as to how they need to go, where they need to go next to better their knowledge and understanding. Now, one thing I do find is I abbreviate a lot of my feedback because I have quite big classes. And if I spent all of my time writing very long comments on my students' work, then I'd probably spend very little of my time doing other things. So I find myself using codes, abbreviations, and these are two very common ones that you might have heard of. So when a student provides me with a piece of work, I might write down www and then 2 or 3 points. I might even just put www and then an arrow pointing at a section of their work. So they know that I recognize this is very good. EBI is more important because you could write down EBI and then underline perhaps a command word, or you could put a little circumflex to show whereabouts they're missing a key term. Very quickly, your students will understand the shorthand you use. And let's be honest, they're very used to using shorthand. This just means that feedback that you provide is both of a good quality, but also that you can deliver a sufficient quantity over the course of the IGCSE. Again, how do the Cambridge resources support us as teachers to deliver formative assessment in our teaching? Well, there's lots, I must admit. I think probably even more than the old editions. The new editions place an even greater emphasis on formative assessment and also peer assessment. which is very, very closely related. So here is another sample spread from the course book. You'll see that we have over here a series of questions, which are traditional questions that you can find the answers for later in the book or elsewhere. These can be used perhaps as starter or plenary exercises. And of course, what you can do is you can have a look at the students' responses prior to the next lesson to decide whether you need to go back and revise and review, or whether you can simply push on with the next section of this topic. Over here, however, is something a bit more sophisticated. Over here, this is directed at learners themselves. So I'll just read you the first point here. It says, functions of the parts of a plant cell. You could work on your own or in a pair for this activity. You will need, and there are a couple of items, and it guides students through how to draw, how to label, how to explain what a good diagram is. Down here, we have a section on peer assessment, and it helps students understand why peer assessment is useful, but also it helps them very, very explicitly to give quality feedback to their peer. And in my opinion, helping students to appreciate why peer assessment is important encourages not only quality formative assessment, but also bolsters and strengthens the active learning techniques we referred to earlier. It's all about encouraging students to engage with each other. Of course, you will have to get involved as well. It may well be that one of these peer assessment activities you model. So you pretend that you are a student, you ask for a volunteer, and you engage with that other student as if you were, again, a member of the class, just to show them how it's important to be honest about each other's work and not just focus on perhaps some of the superficial things that students often comment on. The workbook has examples of self-assessment. And of course, this perhaps is even more important in the times we are living in. So we have here, for example, a spread from the workbook that requires students to draw and label plants and animal cells. What's important here is that the workbook guides students through how to mark their work out of a certain subtotal. And as you can see, these are the very familiar points that are required ultimately when students undertake the examination. Skills in diagram drawing, vital to this subject, and learning them early and being able to develop them from an early stage is vital as well. What's quite nice in the workbook is that you will find examples of students' work. So here we have two examples of leaves that have been drawn by two students. And it's quite nice because the students themselves, the ones that you have in your class, are invited to comment and criticize and also praise what these imaginary students have produced. So this can, again, facilitate work undertaken at home. And providing success criteria in this way, you can see that the diagram on the right-hand side is very, very good, can illustrate very powerfully to students how not only formative assessment is useful, but also how they can excel themselves with their own work. The skills workbook, here we have an example from the maths workbook. And here we have an example from the English language workbook. Lots of resources available, but as you can see from these sample spreads, all of them place a vast emphasis on formative assessment, showing students how to do things, encouraging them how to do it themselves, and then providing them feedback, providing examples, success criteria, modeling how things are done. This is all good teaching practice, and it's there in the materials. Okay, so again, given the short time we have together today, we're now on our third of 4 short sections. I'm delighted to say we will have some time for questions at the end. So if you're sitting there thinking, I really want to ask this question about something I've said already, we will have those opportunities in about 15, 20 minutes time. So how do we go about planning a lesson, an effective lesson for the new IGCSE Biology? Like I said, we'll have a quick look at how the new CUP books can help us, not only in an active fashion, but also how can we assess them using the books. What's important for me to say at this stage is that there is no expectation, if these are the course books you are using, for you to cover everything and use every activity. They are meant to be a bank of suggestions from which you can select the ones that are perhaps most aligned with your own teaching practice. Okay, so planning a lesson, the very first thing I would do, and I do when I plan my lessons, is I go to the syllabus. Okay, so I go to the very first document I showed you today. Let's imagine we're planning a lesson on transpiration. Transpiration, my own students sometimes find a little bit dull. Anything to do with plants, I always find that students need a little bit of convincing of its importance, which is a shame. But I do find that once we get started with transpiration, they do enjoy it. It's also quite a challenging activity because there are a lot of key words, as I'm sure you know, which students find very difficult to distinguish. So here is what we're going to be looking at from the syllabus. I would then spend a moment thinking to myself, what do I want my students to come away with at the end of the lesson? So I need to identify my learning objectives. So I go back to the syllabus and I look for patterns here. I look for ways in which I can group some of these dot points together to tell a story over the hour or 2 hours that I might have with my class. So I might group these two together because that is a fairly fundamental part of the early lesson. I need to make sure that we have revised the importance of water in plants and provide them very early on with a definition of transpiration. The mechanism will probably be my second focus of the lesson. How does transpiration occur? And then we will have a look later in the lesson at the factors that have an impact on its rate. With regard to learning objectives, I would suggest that probably globally, myself included, until probably 2 or 3 years into my teaching career, I wasn't really very confident in what made a good, measurable learning intention. Think about what you want your students to do, not what you want them to know. And they have to be measurable. So what I mean by that is not a particularly good learning objective. I want my students to know about transpiration. Not only can you not peer inside their brain and find out whether they know something, that sounds like a very different world, but also that's a very vague statement, isn't it? What does that mean, knowing about transpiration? Much better is to say, I want my students to be able to state the name of the pores in leaves, describe the process of transpiration, and explain the mechanism of transpiration using the word cohesion. They are measurable, they are clear. And one thing you can, I hope, spot is that the last three there are using Cambridge International command terms. And I would encourage you to do that. I would encourage you to use in your learning objectives the Cambridge International command terms to encourage students to distinguish between them. Those three on the board there, by the way, state, describe, and explain, are very often misinterpreted, particularly the last two. And I want to share with you a very, very, very simple activity on this point, which you should try with your students just to help them distinguish those command terms. State, describe, explain. Well, here on the left is the flag of my country, the United Kingdom. And on the right, I'm sure you probably recognize that one as well. That's Japan. That's the flag of Japan. Now, if I said to you, state the country for which the flag is representative, then you would probably say, I hope, United Kingdom and Japan. If I asked you to describe it, or my students to describe it, well, I would expect many of them to venture into the idea of talking about what the symbols mean, and that is incorrect. They must simply talk about what they can see. So a description of the flag on the left would only mention the colours and the shapes. It's the what, it's not the why. And in biology, a lot of students accidentally stray into talking about why when they should be talking about what. To explain the flag on the left, we would have to talk about the fact that the central cross represents England, the blue comes from the flag of Scotland, etc. But we are using the word because when we refer to explain. And it's very important in biology, as I'm sure many of you know from your past experience of marking students' work. These two command terms couldn't be more different, but students often do mix them up when they are asked to do one or the other, either in a practical task or in an exam situation. So over here, of course, this would be Japan. That would be the answer. Description would be a red circle on a white background. The explanation, oh, I should have researched that before I spoke to you today, but we'd have to talk about what this flag represents, and we would use the word because. So having determined your learning objectives, now we know what we want them to learn. How are we going to teach them this? Well, the first thing is, I would always say, within the first 5 minutes of your lesson, you must do 3 things. And it should be fast paced. I like my lessons to be quite frenetic, to be quite almost stressful for the first 5 minutes, not only on my part, but even the students. I like to get the adrenaline going. Either it's the end of a long day and there might be a little bit of misbehavior in the air, or it's the very beginning of the day and they need a little bit of a shock to get started. So I will make sure that there is some hook or enthusiastic starter activity which encourages them to get into the mood for a little bit of learning. And we'll talk about an example of that in a moment. But also I want them to provide me with a little bit of information. Not only do I want them to get into the mood for learning, but I also need to find out what they remember, possibly from my last lesson. But on this occasion, transpiration, we're going to assume it's the very first lesson. So I want to find out what they might know about plants from their wider reading, maybe from Checkpoint, if that's what they did prior to the IGCSE. And then I will share with them what we're going to be doing today. So how could the resources support this process? Well, as I said to you earlier, every chapter begins with a contextual story. And one of the nicest contextual stories relating to transpiration is how on earth do the biggest trees on earth take water to the very highest leaves? So I would show them some of the giant redwoods that grow in California, with the scale of a person next to it. And there is a very nice image somewhere of a tunnel that's been carved into one of these trees and a car is driving through it. I would wow students with the idea that these trees are able to transport water at the rate of gallons an hour in a complete silent way every single day. Get them hooked, get them interested, get them to think. That throws up lots of questions. I would then have a general discussion with them. I might direct them to perhaps one of the images in the textbook and ask them to explain what they know about water's role in a plant, to encourage them to explain to me what they know already. And that will give me a very, very subtle indication as to where I need to pitch my lesson going forward. Later in the lesson, of course, I might undertake some practical work but that would be supplementary to the introduction. I might mention that. So a good lesson, in my opinion, needs to be broken up into several chunks. And in previous years, decades, people have often talked about the tripartite lesson, which is a starter, a main body, and a plenary. That still works for me, but I do find that sometimes my starter activities end up becoming 15, 20 minutes long, just because I need to find out a little bit more about what my students know, or we have all got a little bit carried away talking about the contextual hook. It's very nice to have that little bit of flexibility, but having these chunks of material, having these chunks of activities can be very useful because it adds variety to the lesson. The textbook, the workbooks, clearly by their very nature provide lots of opportunities for you to have these 10, 15, 20-minute discrete opportunities for learning. Sometimes they are a little longer. You'll see here that there are some very, very clear guidance provided in relation to some of the practicals on the transpiration story. And then at the end of the lesson, you should always ensure there is an opportunity to review students' knowledge, not only for you to have a much clearer idea of their current knowledge and understanding, but also it's nice to help them leave the room reassured that they have made progress during the last hour. And never underestimate that. Make sure that your students appreciate that they have covered part of the journey during the last hour or two. Conscious of time here, so I'm just going to flick through a few of these slides, but you'll see in front of you there's lots and lots of opportunities for active learning, formative assessment provided by the workbook, provided by the skills books. Okay, we have about 5 minutes left now, and I want to spend the last part of this session on something which, you know, a year or two ago we probably wouldn't have given much thought to, but how do we encourage students to work well at home, including opportunities for practical work? Well, I've got 2 or 3 tips, and all of us are in a similar position. You know, I mean, very few of us this time last year had ideas to encourage ownership and encourage students to take more steps to become independent. But now I'm sure that you could probably teach me a few things about what you do with your students. So these are just my own ideas, and these have worked well for me during the period of time when Singapore schools were locked down. I found that the following guidance would have been useful for me to hear. So if students are working at home a lot, then it is important to encourage them to take ownership of their own learning, set their personal goals, and engage in a lot more self-assessment. Now, like I said to you earlier, the workbook in particular from Cambridge University Press does have a lot of this type of activity. So plenty of opportunities are provided which walk students through how to undertake self-assessment with, as you can see at the bottom, a little bit of guidance to provide them with an idea of how they're doing. One thing I would encourage you to do is to always press your students to become reflective even before they begin a topic. So before they start learning about transpiration, for example, get them to ask themselves that question at the bottom. What do I already know about this topic? And the materials in the textbook and the course book and the other publications are very useful, because at the beginning of each chapter, there tends to be a series of dot points that suggest to students where they're going. It doesn't tell students what they'll be learning, but it gives them an idea of what they'll be doing. So you could ask your students to read down this list and maybe annotate each of those dot points with any keywords they've heard of, or maybe any definitions they're aware of, just to encourage them to link what they know already with where they're going next. Online questioning, I've found, has been a real problem in the past year. What I found works well for me is that whenever I've hosted classes online, planning questions I would like to ask my students has been very, very effective. So we're all used to making lesson plans, but as part of those lesson plans, I've actually included 2 to 3 questions that I know I want to ask. And sometimes I've actually identified who I want to ask them of. Make sure you give enough thinking time to your students, even online. It can seem a bit strange when you ask a question and it's very silent for 5 or 6 seconds, but lots of evidence suggests that teachers don't give enough time to students to think before they give an answer. Try answering questions with another question just to get discussions going. So if a student asks you a question and you provide an answer, extend that answer. Maybe ask them something along the lines of, how could you apply this information to another situation? Or where have you encountered that word before in the course? And don't underestimate the power of varying your ability to use open and closed questions, even online. Now, we haven't really got time for this, but if we had an extra 15 minutes, I would take you through this activity and we would engage together. But it's worth thinking about. What I would ask you to do is consider that the possibility of me thinking of a cell type. If I was able to provide you time to ask me questions, we would do that, but I would only be able to say yes or no. It would take you a long time, I think, to find out the cell type I'm thinking of. But if I gave you the opportunity to ask me open questions, you would get to my cell, you would guess my cell, I think, a lot quicker. And have this discussion with your students, be open with them and say that teachers do have the opportunity of asking either closed or open questions online, so that it really clarifies to them how they can ask you more effective questions to get their answers from you. And we are working at a level here which is going into metacognition, and that's not something I want to speak about today. But being open with students about how we teach them can better the experience for them as well. Okay, and finally, the other thing I found last year when I was teaching online was how to provide opportunities, meaningful opportunities for students to engage with the IGCSE in breakout rooms. So I would often say to students, right, I want you to go away and work as a small group on this question. And I might give them a past paper question to do, and then I would go around the breakout rooms and listen to what they were discussing. I found that wasn't a particularly effective way of encouraging discussion among small groups. What's better is if you use some of the following word games, and they really do like these, and they're quite nice 5-minute activities. Of course, these can be done in class as well as online. But what I might do is at the end of a topic, I will give students 3 words. Let's take sperm, pollen, ovum, and I will encourage them for 5 minutes to discuss how can you make each one of those the odd one out of that list. So many students would say, oh, the ovum is the odd one out because it's the only female gamete. But then you could say that the pollen is the odd one out or the pollen grain is the odd one out because it's a plant gamete. But then you could say maybe that the sperm is the odd one out because it's the only one that's able to engage in locomotion. And the discussions among students can be very enlightening for each other. And it's showing you, if you're listening to these discussions, whether they really do understand a topic, because they're making links and they're addressing each other's misconceptions in real time. There are other opportunities which we probably don't have time to discuss now, but asking students to discuss in a small group and decide on a small paragraph that includes every single one of those key terms in context. Or maybe playing this game, which is a very, very nice one. which again is fun and can be fascinating to listen to if you're in a breakout room, getting students to discuss the meaning of photosynthesis without using light, glucose or oxygen as key terms. And that's the point, as said on the screen. You're able to listen rather than speak. It means that it's not all just about you. It's not a monologue. You are able to listen and gauge students' understanding, even online. We are very, very short of time here, but I just want to mention one last thing. Do encourage communication with your students, not only between you and them, but also between each other. There are lots of wonderful free sites out there. I've listed a few on the screen here. I encourage you to try those and others to encourage your own students to engage with each other if they are isolated at home. Practical work can be done in a home-based setting. I think many of the publishers are looking at expanding this going forward, but you'll see that practical work and its qualities and its purposes that we all know and love can be encouraged from the comfort of a student's own home. In biology, just get them to go outside and do some measuring of leaves and plotting graphs and just get some fresh air. But you'll see that within some of the practical workbook pages, there are lots of other activities that students can do, assuming they have maybe a ruler at home or a piece of wood. They could do the human response reaction time test, which provides a whole host of quantitative data that they can process. There are lots and lots of other things I'd like to talk to you about. Cambridge Resource Plus is a lovely platform which is free to use during the pandemic, Cambridge Resource Plus, and that is a good source of very high quality video tutorials that Cambridge has produced that help students appreciate some of the results from food tests, for example, that they probably wouldn't be able to see if they're working at home. So have a look at that at some stage. It's a very, very nice and free to use resource for the time being. Okay, what I'll do is I'll just leave these final tips on the board. I won't say anything about them. I think they're fairly self-explanatory, but these are the last tips I have to offer you if, and I hesitate to say it's going to be for some time, some of you are still teaching online. These are things that I found have helped me over the past 12 months. And I hope that you're able to take a screenshot and have a think about those, and maybe it might, some of those might help you going forward. So conscious of time, everybody, and not wishing to take up too much of the time we have at the end, I'd like to now pause and ask Tamsin to come back. We have some time now for questions. And just at this juncture, I'd like to say thank you very much for listening to my monologue. And I look forward to maybe meeting you face to face in the future.
Wonderful. Thank you so much for a brilliant session, David. We had a lot of questions that came through about remote teaching. So let's pull those thematically together. So the first question is, how do we make learning active when online? How do we do practicals when we're teaching online?
Okay, so quite a few of the strategies I mentioned throughout the screencast today, they are 5 to 10 minute pursuits. It depends really on your confidence and your ability in hosting breakout rooms. Active learning for me involves students working together, and that is possible at home. It's just a bit more difficult for us to manage as teachers, and we probably have to set our standards a bit lower in terms of how we're managing behavior and how we're managing the level of focus. We have to trust our students a little bit more in this context. So for example, if I was setting up a breakout room where students go away and engage in some of those keyword activities, or maybe work on an extended exam style question, a 5 or 6 marker, then I would encourage students to have key roles in the breakout room groups. So I'd have somebody that perhaps could be the scribe. They are the person that will explain to me when I visit the room what the state of their answer is or what has happened so far. But then I might give another student a different role. They could be the recorder. They could be somebody that makes a note of problems they've encountered. It does require a bit more planning. I do feel that my teaching back in class has actually improved from the experience of teaching online last year. So if you're thinking, as we all did when we are teaching from home, I hope this is over soon. Do rest assured that a lot of teachers, when they go back into the classroom, not only feel, wow, I feel far more confident in managing a large group of students who are working disparately, but also I think it's a case of, well, I'm so pleased to be back again. So in the case of the practical work, I mentioned Cambridge Resource Plus. I mean, if you type in Cambridge Resource Plus into Google, it'll be the first hit you get. That's a really good source of, like I said, videos, which take students through how the practicals are done in the lab. There are lots of interactive, free-to-use platforms out there. I'm sure many of you will have heard of FET Colorado. It's not affiliated with Cambridge University Press, of course. I have to make that clear. But it's a very, very widely known platform, which is open access and free to use. And that does have a lot of biology, increasingly some biology demonstrations on it. There's also quite a lot you can do with YouTube. There's lots of time-lapse activities which show plants growing or show food in transit through the digestive system. And with a bit of imagination, you can get students to plot graphs using that. They can pause the YouTube video at times and they can plot graphs to kind of show how things progress in biology. So I'm afraid there isn't a platform out there yet, and I'm sure there will be after this pandemic, but there isn't a platform out there yet for students to go away and do experiments online, and simulate a full experiment online. So we have to use our imagination a little bit. And the same is true of active learning when students are at home. I hope that half answers the question.
Brilliant. Thank you, David. Thank you. Now, the next question is about teaching in the classroom. So how do we handle learners who are taking more time during an active learning session, especially when the sessions themselves are often timed?
Okay, well, it's very, very dependent on what the activity is, of course, but I would always say you need to make sure you know your learners as early as possible in the course. And if there are learners that you know are more inclined to struggle or take a longer time to get started or even progress under active conditions, you could ensure that they have a buddy to turn to. Maybe if they're working in a small group, they've got somebody they can lean on, they can seek guidance from. You could alternatively put some of those students together in a small group. I mean, active learning isn't always about group work, but often it is. And then you could associate yourself more closely with that group of students who require a bit more guidance and your time. So again, it's about careful planning. And we're very used as teachers to planning lessons from a content point of view, making sure that we've read the material. But I would say that, even myself, acknowledging this as a weakness of myself, I probably don't put as much time as I should into planning the interpersonal interactions that happen during my classes. That's something I neglect and I should do more of. I should sit down before a lesson and think, right, I want those two students to work together during this 15 minutes. And during this 15 minutes, I'm going to be in that corner of the room. I mean, that is expert level planning, which often we don't get time for. But if you're able to do that now and again, then I think that you will get into a habit of doing it even without realizing it, and subtly planning your lessons on that basis as well. Thanks, Tamsin.
Brilliant, thank you. Now we have a question about marking schemes, and it's about keywords. So when learners are writing an answer to a question, but they don't include the keywords in the marking scheme, but the answer is still right, can we award marks, or should we stick with the marking scheme?
Yeah, that's a very good question. I mean, the marking scheme is almost like a different language, isn't it? And you have to learn the language of the marking schemes to fully appreciate how to apply it. So the general rule is that if a word is underlined in the marking scheme, then that word must be included in order to credit that mark. It doesn't actually have to be spelt correctly, as long as it's phonetically similar. I would never admit that to my students. I don't want my students to spell photosynthesis with an F, but actually Cambridge is aware of the fact that for many students, English could be a third or fourth language, and we don't want to penalise poor use of the English language over the science. So if it's underlined, then it must be included, spelling notwithstanding. If it's not underlined in the mark scheme, and the student provides, let's say, a definition of that word, or they allude to that word in a very clear fashion, then it does come down to teacher or examiner judgment. And I would say be a little more harsh than you need to be during the course to continuously drive your student standards higher. So it really is very much dependent on the question, but I would say no, it doesn't have to be there if it's not underlined, but you must be convinced that that's what they mean. Thanks, Tamsin.
Wonderful, thank you. Now, another question about active learning, but I guess the challenges of today. In a situation where you have a single student, how do you help him or her learn actively, since peer learning isn't possible here?
Wow, I'm very jealous of the teacher that has one student in their class. I'm sure that could be a situation where the student is being tutored or possibly being taught in isolation for some other reason. If that's the case, then maybe you have to be their peer. So you could be the individual who, in one context, you will be the person who is providing the information. This is what we are doing for the next 15 minutes. But then you would almost have to put a different hat on, so to speak, and then pretend to be somebody who is in their class and have the discussion as if you were a fellow student. If you are in a classroom with that individual, then it's possibly a little bit more difficult, because separating yourself from being a teacher could be more challenging if you are doing a practical with that student or building a model of a cell, but it is entirely possible. And like I said to you earlier, being active doesn't always mean being social. Answering exam questions is actually active. Of course it is. Students are doing things. What's important, though, is that there is an opportunity after that period of individual activity where there is a discussion, where there perhaps is a proactive nature on the teacher to identify some of the weaknesses in the student's work, and then have a discussion with the student as to how they can improve their work. So yeah, I don't want you to think that active learning always has to involve talk or building or model making. It doesn't. It just involves doing. That's the key point. Thanks, Tamsin.
Fantastic, thank you. Now, another question, this one about formative assessment. Basically, can you explain more to us about formative assessment and how you might use it?
Okay, so yeah, good question. I think I'll give you an example from a lesson yesterday I taught. So yesterday I taught a lesson on, in fact, it was an A-level lesson. We were looking at protein synthesis. And in order to move on to the process of translation, which is the second stage, I need to be reassured that students have fully mastered the first stage, which is transcription, because the product of transcription, as biologists will know, mRNA, is a vital part of second stage, which is translation. So what I did at the beginning of my lesson is I spent 5 minutes having a missing word exercise. So I provided the students with a block of text with missing words and a few mistakes that had been made intentionally within the block of text. So they had to fill in this block of text with missing words about transcription. I then asked them to provide those to me very quickly. I flicked through them. It took about 2 minutes, during which time the students got themselves ready for the lesson. I noticed that quite a few of the students made a very, very common mistake, which was that they identified, they'd written down that thymine was in the mRNA sequence rather than uracil. So we just spent a moment then discussing why that mistake was very common and also how they can avoid making it in the future. And without that little bit of formative assessment, which only took 5 minutes, we would have all started with a bit of a misconception, which would have come up at some stage during the lesson and, you know, derailed the progress. It would have meant that we would have had to have stopped, rewound, and that would have taken away from the direction of the lesson. So having this opportunity at the beginning of a lesson, just briefly to have a review, and then for you to have a look through responses is vital. My class of A-level students is only about 15. If it was larger, and my IGCSE class is larger, I might ask them to do something on a mini whiteboard or give them a piece of blank paper, and they can hold that up and I look around the room to see if they've identified a key part or label, or they've drawn an accurate diagram of something before we move on with the next part of the lesson. So yeah, that's not the only type of formative assessment, but that's the first one that popped into my head because I used it yesterday.
Fantastic. Thank you, David. Now, a question about the lesson guide. What are the main features of a lesson guide?
Right, lesson guide. I'm going to have to sort of guess here as to what that means. So lesson guide in terms of plan, or maybe the teacher's resource. So I'll take it as meaning that the lesson plan. So lesson.
I think possibly the TR we're talking about here, the teacher's resource.
Oh, very good. Okay, so the teacher's resource, yeah. Well, it took me a whole 12 months to write that, so I know it very well. It's very detailed. I mean, you're probably thinking, well, of course he'd say that because, you know, he wrote it, but I think it is very good. It's the sum total of really my career in biology teaching so far. So about 15 years worth of experience teaching IGCSE. What you have at the beginning of every chapter is you have a list of the most common misconceptions that students encounter and sadly often never overcome, with suggested activities that you can use to help them resolve and clear them. It goes then into some suggestions for starter activities, many of which are active and many of which also use the course book. Then it goes into main body activities, all of which are about 30 to 40 minutes long. Then there are opportunities provided. You can select from 2 or 3 to summarize the lesson under a subtitle of plenary. You've then at the end of each chapter in the teacher's resource, you've got guidance related to the project that you'll find in the new course book. So the new course book has a section at the end of each chapter, which is a self-led project that students can do either individually or with each other. And the teacher's resource provides extra guidance on that. There are also links to online resources, which are not Cambridge University Press. They are beyond the publisher. So these include things like the World Health Organization. And there are other ways to help make your class learning more contextual. So, I mean, the thing that I find would be the most useful, I mean, it was most useful for me, was actually the misconceptions list. It is very detailed. I can't think of any other misconception that students would have for any other topic. So if you do have the opportunity to have a look at the teacher's resource, I encourage you to try it and get in touch with Cambridge with any thoughts and queries.
Brilliant. Thank you, David. And now we're moving into the last 5 minutes of questioning. So I'm just going to say we probably won't be able to cover all the questions here, but what we're going to do is we're going to create a blog post that we're going to put on our website and email out to everyone, which will cover all the answers for the main themes and the questions. So we're going to answer everything we possibly can now. So let me see. The next question is on comprehension. So how can we help students who struggle in comprehending biology terms?
Okay, yeah, good question. It is a very good question. So that question is important in my own school here. We have a lot of students who have recently moved from China to Singapore. And for them, English is a challenging language to learn. And biology, of course, is one of the subjects which has probably the most new key terms to learn all at once. Somebody once told me, well, two things. The first thing is the longest word in the English language is biological. It's the name of a certain disease that you get when you inhale, I think, dust from volcanic lava. And the second thing I've heard, which I can't prove true or false, is that in a typical biology degree, you actually have to learn more words than in the first year of a typical language degree. I mean, I can believe that. Biology does have so many new key terms. So the way around this, and again, there's no easy answer, but I help my own students with this problem by giving them a small vocabulary book, just like they would have if they were learning Indonesian or German or French. I would encourage them to write down every single biological term that we encounter with the equivalent term in their own language, whether that's Mandarin or Arabic, etc. But also a very, very quick description of what it means. So they have this little vocabulary book to accompany them throughout the 2 years. The other thing that's quite nice to do for students who find these terms difficult is the activity I showed you earlier. So I just want to quickly flick back. We didn't have time to look at this one in detail, but that's this one here. I've called it What's the Story? But the point is, if you give a student the key terms and then ask them to produce a short paragraph using those, or at least some of them, it removes what we call the cognitive load of having to recall key terms. Now, I know that's not something we want them to forget about in the long term. They must know these key terms, but it does help them develop some of the higher level skills which are listed in the syllabus under the subtitle Assessment Objective 2. So this is application, synthesis, all of the things that you will find at the top of Bloom's taxonomy. Often students for whom English is difficult will be locked out from accessing those higher level skills because they've forgotten the key terms or they can't use them. So providing them with the words now and again boosts their confidence in using them and feeds into this cycle of positive progress. They are then more minded to sit down and learn the terms if they can see what they are able to do with them. Thanks, Tamsin.
Fantastic. And can I also mention that we also have the glossary at the back of the coursebook, which is incredibly useful. And we are publishing an English language skills workbook next year, and that's specifically for Cambridge IGCSE Biology. So it focuses on language exercises in the context of the actual syllabus, which should prove very useful. And you can find out about all of these on our website, cambridge.org/education/science. Now, I'd like to finish with one final question, because I think this is really important, because you often get even able math students who struggle in a science classroom. And so could you get some help for some important points to diagrams and graphs?
Okay, yes. And I'm glad you mentioned the English skills workbook there, Tamsin. There's also a math skills one which relates to this question, which is published for this specific syllabus by CUP also. So yeah, graphs, tables, diagrams, these are a perennial, annual source of frustration for many a student. And I guess the thing I would say is that reassure your students that the rules remain the same. So as long as they are able to produce a graph of quality today, that won't change in 2 years' time. So it might be worth you actually devoting some of the early part of the course, the first part of your IGCSE journey, to mastering some of the skills that crop up quite a lot. Making sure that diagrams are accurate representations of an observation, no shading, there's no shadow in an actual specimen, sharp pencil, no overlapping lines, these things. Yes, some students find it difficult, literally with a pencil, in terms of tactile ability to produce them, but knowing the rules are important, and they can then practice over their next 2 years of the course. Same thing with graphs. Again, there are rules. The independent variable must be on the x-axis. There must be a figure, whether that's 0 or a number higher than that, at the origin. Unless otherwise stated, the points on the graph ought to usually be joined with straight lines. These rules are out there. They're in mark schemes, they're in the Cambridge University Press materials. I feel that what often happens is that teachers, including myself, we probably leave a lot of the skill development for paper 6 towards the end of the course. And if we introduced some of these skills early, and you'll notice that some of the early chapters in the materials I showed you earlier, for example, cells, there are opportunities to draw and plot and label. So doing this earlier, I think, will boost confidence. And I find that it's more of a confidence issue than an ability issue when it comes to skills such as graph drawing and diagram drawing. Thanks, Tamsin.
Brilliant, thank you. And so it just remains for me to say thank you so much, David Martindill, for your fantastic presentation today. And thank you everyone for attending and contributing so many interesting questions and responses. As I said, we will be creating a blog post with answers to the questions that we weren't able to handle today, just because we had so many good questions that came in. So thank you everyone who didn't get their questions answered as well. And thank you so much for attending today. Remember, 7 days after this webinar, we'll send you an email thanking you for your attendance and containing a link to the recording. And to find out more about the resources discussed today, please speak to your sales representative or visit cambridge.org/education/science. Thank you, everyone, and goodbye.
Goodbye.